And I know that two of our panel members have to catch a plane to Southern California. So this part of the agenda was for panel members to make summary comments. And so I guess I would ask Sandy and Ed to speak generally if you have anything to say.
So I'll just summarize. I want to say that I think this is really great work, and I want to just reemphasize what you're already doing, which I think is great. And so I think randomization is a key strategy for doing a sampling, and I think to try to stick to that as much as possible. I heard about the challenges that there are in actually getting fields to agree to participate, and I think keeping track of what factors are sort of associated with fields not participating could help you understand if there are any biases that might be popping up if, for example, private field owners weren't participating as much as public field owners or so on. So I think trying to keep the sample as random as possible, but understanding sources of non-randomness would be good. Again, the idea of replication that came up a little bit in the panel discussion, wherever it's possible to replicate samples, not only within sort of sampling strata, but replicate within the same field. I think that's a very nice feature to try to do whenever possible. I heard a lot of discussion about trying to control for other confounding factors. I want to, again, encourage you to do that if you're, if possible, to sample at the same time of day or to control other sort of factors that could confound the results. Again, as a statistician, I love big sample sizes. So whatever you can do to use resources efficiently to try to get the largest sample you can to try to, because it is a highly variable exposure with a lot of different factors of interest. So use your resources as best you can. And then I just wanted to make another comment that I heard a few comments that were relating to the fact that the factors were categorized, and maybe then in the analysis you can use the quantitative values of the variables, and I think that's a good point as well. But certainly for doing the sampling, you need to have sort of categories of strata is just a common sense way to go about doing it. But in the analysis phase, if you're trying to relate the levels of some chemical to ozone, you could certainly use the quantitative ozone values if you did measure those at the sites. So that's the end of my comments.
Thank you, Dr. Eckel. Ed.
So I think it's a very impressive set of work that you've laid out on and that you presented today, and appreciate all the hard work. Again, I would say that one thing that we didn't talk a lot about today, but which I think you need to think about, is how this data is going to be used, how it's going to be interpreted, and what are you going to do with it? As academics, we get excited about any individual experiment and collecting data, and every piece of data is useful in raising other hypotheses to address other issues. But I think from the public standpoint, we need to think about how this is going to be used and interpreted. And so in that sense, I think making some decisions such as we talked about regarding the types of field, the number of selectionable regions based on climate, et cetera, may have to be, in a sense, compromised in order to get data that you can actually interpret to make some judgments later on. I think you've gotten some amount of feedback throughout the course of the day, and I look forward to hearing what more is going on. And we made a number of specific comments, so I don't think it's worth revisiting all of those.
Thanks, Ed. Where do you want to go next?
So I want to emphasize just a couple of things. One, I think it was Dr. Madalena's slides that had something to the effect, we don't want to be astonished by surprises, or we're going to be prepared not to be astonished by surprises and be flexible. I think that's very important. There's going to be a lot of things that don't go well. And I think, again, I would add it's a really well-organized exposure study. And then basically to second the point that we have to remember this is not, it's a great exposure study at this point, but it's part of a risk assessment. And we haven't yet seen how all these pieces are going to fit together. So we have to keep our eye on the bigger question. And, we're going to then struggle with, hopefully we're going to get just really reliable representative exposure information, but we're still going to have to figure out how to put that in the context of the broader question of health risk. So unfortunately, we can't just walk away and say we got a great exposure study without thinking about the integration with the overall health study.
Dr. Bennett?
My thoughts were kind of similar to that, especially when we were going over and identifying all these new compounds that we weren't really sure what we were going to do with. I think it's not going to be too early to start thinking about what we can do in terms of toxicology, like what fraction, I know the EPA has a lot of high throughput toxicology data, what fraction does that space even cover versus what we're finding? Is there any way to work with them to at least do some screening level, some of their high throughput toxicity on some of these things we're finding, and trying to figure out how that's going to be handled, because it seems like we're going to find a lot of new chemicals that we don't know very much about. But other than that, the exposure side looks great, super thorough, and the lab work, it seems amazing. So it's all very impressive. But my only comments are on thinking ahead.
Okay, my comments are sort of like Ed's. You've got to keep your eye on the ball of the goal, and I think that as you listen to the public comments, it's really, is there a risk for my kids to use the field? And I think that I had said it before, we need to, understand some general cases, but we need to make sure that we're looking at some of the high-risk cases just so people can say, you've looked at those places that we think are the biggest problem. We need to understand what is coming from the crumb rubber, because in fact, that means we are going to need some background, and I think that was a very good suggestion. Again, what's in the crumb rubber, what's in the exposure samples. I said it before, we need to work with other groups because all of these studies are so expensive. We're going to have to start developing common protocols so that as more data comes out, the body of the data can be used in assessing the risk assessments. And one other thing. Oh, and once you get to the risk assessment, you need to keep the exposure pathways separate because chemicals that are inhaled can be transformed by the body and go to a different target than chemicals that are ingested. And so you just can't say, well, here's the sum of PAHs. The highest exposure for PAHs is usually ingestion, eating food that you cook. But I think that goes down to the risk assessment. But when you do the risk assessment, don't lose sight of all the specifics there too. And I think you guys are doing a great job.
I'm going to take the chair's prerogative of seconding something that you said, because it was going to be one of my main comments. I really think that we have to work closely with our federal partners, if they're going to be able to go forward with their planned studies, for the reasons that, Dr. Sheldon pointed out. I think it would be really very unfortunate if different methods led to different conclusions and the public would be confused. That would be, totally against everything that you're trying to do so well here. Dr. Kyle, I'm sure you have some comments.
Yes, thank you very much. I'm happy, though, that we're all sort of on the same page in the comments here. So that's sort of a warm feeling for us, isn't it? So in terms of pulling this all together, I think there's a lot of pulling this all together kind of comments here and explaining it. And I would add a couple of ways of thinking about that are different than what we've heard. One is to think a little bit more about the phasing of what we're doing in terms of what questions are being answered in what order and how do the different streams of this inform each other. Because I'm not quite sure we've got that yet, or maybe we can't quite see that. But there are parts of each phase of the field work that will inform different things. There's a question to me about whether the biofluid stuff should be done sooner or later, because what if we find that even the levels without doing that are not of concern, then you don't need to do that. So, there's some issues like that for me. How do we set this all up so we can explain what we've answered? How there's the sort of the research questions and the thinking through of it part are missing. There's a lot of how to measure stuff, but not much on how to think about what it means. And I think that really needs to be more explicit here. We call that hypotheses, research questions. Maybe it's data input that we need for a risk assessment, but that can all be elucidated a lot more, and then that would help us see whether we've got enough stuff. Like other people, I'm worried about the tox data for the risk assessment. You know, if we end up with all this exposure stuff and then we have 700 untested chemicals, where did we get with all this? I don't want to say nowhere, but it won't be towards having a clear answer. So I'm wondering when we're going to get to that, because that's kind of where it looks like it's headed to me. I'm exaggerating for effect, but you know that it looks like there's a lot of stuff here where we don't have much data about the toxicology side, and there's no epidemiology either. And then this question of the cumulative impacts, aggregate exposures, all that. That has to be dealt with overtly. We can't. I don't know what we think about that. Are we just going to treat everything separately, like most of these other assessments have done, and ignore all the other exposures, or not? I think that's important. But I won't go into what to do about that at this moment. Thank you.
Well, again, I'll take the chair's prerogative to have the last word. First of all, I want to thank everybody here, the panel, the OEHHA staff, the Lawrence Berkeley National Lab collaborators, the public. I think the public comments were very astute, and I know there are many that we haven't yet addressed from by email, but we'll get to them too. You know, one thing I like about California, I'm on the California Resources Board, and our public hearings are, you know, there's a lot of dirty linen that comes out in the public meetings, but you know, in California, we pride ourselves in having open meetings, especially in the environmental health arena, and this is another example of this. I really think everybody did a lot of hard work here, especially the staff and the Lawrence Berkeley scientists. But I agree with Dr. Kyle that, you know, the end product has to bring these various strands together. You know, we're only partway through the process, and we haven't dealt with step 5 and step 6 yet, that we're gearing up for that. That's going to have to bring the strands together. But I think the work to date is really pretty impressive, and I think I'm speaking for my fellow panel members in that regard. So I thank everybody for good work up till now, and look forward to the weather cooperating to finish phase 2 so we can go on to phase 3.